So this is probably going to be the least mathematical presentation of the whole conference, since indeed I'm not a mathematician, I'm a real biologist. So this is going to be an interesting confrontation, I guess. And yes, at the moment, actually, I teach ecology and also lots about what is science and what is innovation, as I am currently head of research at Antwerp Maritime Academy, where we actually teach people how to sail with big ships. That's our main job. Right, so plants on the sofa. And I had to go back a little bit into my personal history. So there's plenty of references that you will see strewn across the presentation. But we want to talk about plants on the sofa, plants having stress. And my topic of my general thesis is stress is normal. Stress is good. And the way we perceive stress as humans is not the way that we should not translate our human concepts to plants. And the way plants treat stress is probably a better way than what we do. That's the more holistic part of you. So I will first bring you up to speed with what is stress. I think you all know what stress is from an empirical way of life. We all have stress. I will then talk about what types of stress there are for plants. And I will then look at what will probably interest you. I hope it will interest you. That is a set of observations that my colleague Marcel Janssen and I, Marcel at University College Cork, and I did, let's say 15 years ago, or even more, 20 years ago, where we were looking at how plant growth changes because of stress. And now coming back to Weiwei's presentation, there we could only find, at that moment, we could only find linear changes. And now I'm very interested how this could possibly translate into a 3D model. After that, I will again come back and go for what could be stress for a plant's point of view. First of all, what is stress? Well, think of it, you're this fish. You are living in a situation where there's not enough space, where you don't have contact with the other fish, and especially every possible moment you can be killed. Now that's what we all agree, or what we all feel, that is stress for us. We have too much to do. We have there's always the sword of Damocles hanging above our heads that will kill us. Well, we don't like it. Actually, we prefer to be left on our own, to do nothing if possible. Well, problem is, that's also not good. If I go for a very broad idea, then actually we know already for a longer time that a bit of stress here actually is good for our functioning. If we have no stress at all, we're also not going to perform. If there was not the stress of having a deadline for today or last weekend, then I would never have made a good presentation, and so on. You all had that because you all had to make a presentation. We like to be here. We like to be in our comfort zone where it's relaxing and we have our coffee and we do a little bit, but not too much. But still, we know that if we are a bit stressed more, then we can still arrive at more. On the other hand, there is a top point, and if we pass that threshold, we will actually diminish our performance. We will become slower. We will not know what to do anymore. We start to be exhausted, and finally we will break down. This is a more management curve, but actually even that is not a modern idea, because already, and this comes from a paper from 98, but I have to go to the 1930s. There was the Hungarian researcher Hans Selye, who came up with what he called the general adaptation syndrome. And he actually was doing experiments on dogs, where he removed the pancreas and he followed up how these dogs became more and more ill. This is probably an experiment that would not be allowed anymore today. But then he says, well, you have three phases in stress. The first phase is an alarm phase, where we are actually still
Стали проверять, в
not,
голову
not
стукнуло
really
контролирующему проверяющему.
Oleg, could you go on mute, please?
Идут. Самое.
Well done. So we have an alarm phase in which we are triggered by the presence of a stress. We accumulate a little bit of adrenaline. We start to, I mean, like for example, when you have to give a presentation here, first of all, you become inquisitive. You want to know things. Actually, this is still a good feeling. And then after a while, you start to enter a second stage, which is a state of resistance, meaning you are combating the stress. Or you try to acclimate to the stress. You start to make sure that your way of life is in line with the stress. I will come back to that in the very end when I talk about what plants do. And then finally, also even there, Hans Selye said, yes, the third stage is where you can't keep up your resistance, or you can't change enough to acclimatize yourself to the stress that you experience. You will start to collapse of exhaustion and maybe die. Now, that's just a couple of very general topics about ideas about stress. Now let's go to plant stress. And there, a number of years ago, again, Marcel and I were invited to write a paper on what stress is for a handbook of plant stress physiology. Actually, the second edition, because in the first edition, they just had a whole bunch of chapters, salt stress and temperatures and so on, but no one cared to explain what stress actually is. And we said, oops, that's an oversight. Let's first explain what stress is, and let's then start to apply all these different forms of stress. And then we started looking into the literature, and into actually not even literature, we started looking into manuals, and we came up with actually no one knows what has a general basic definition of what stress is for plants. We only came up with this one. Abiotic stress is the primary cause, saying what it is, only what it does, of crop loss worldwide, reducing average yields of most major crops by more than 50%. So actually, that's the basic definition that a biologist or a plant physiologist uses when he talks about stress. It's something that makes my tomatoes grow less good. It's something that makes sure that I have less lettuce on my sandwich. Okay. So we did not agree with that, so we started a whole discussion. First of all, I will give you a few ideas. What is stress? Well, we have two major groups of stress. Again, stress is, according to this very operational definition, something that makes your plant grow less good. And then we have all kinds of abiotic, this is a mistake here, abiotic causes. So we might have a plant that is that has too much UVB light, or a plant that does not have UVB light. If you grow a plant in a greenhouse, they don't get any UVB light. It changes their internal metabolism. We can say ozone, air pollution, of course, light. You all have plants in your houses, I guess. Sometimes the plants do not get enough light. Sometimes they get too much light. That's a stress. You can wound the plant. You can have it too hot, too cold, give it too little water, or you give it too much water. In both cases, your plant will die eventually. An important part when we talk about sea level rising and salt water influx is salinity. There are a number of plants that are very well adapted to be grown on salty soils, and then we have the normal plants that we all eat, like potatoes and tomatoes, and they don't like salt quite enough. We have polluted soils with metals, with all the number of other chemicals. Well, plants, especially when we talk about metals, plants can take up a lot of metals, or plants can start to die because of the presence of metals. And then finally, well, if we don't have enough food, as you will soon notice when we go over into our lunch break, then you will also start to wither away. Well, not die yet, I guess. couple of images to illustrate all that. Well, here you see Arabidopsis. For those who do not know Arabidopsis, it's our lab rat for the plant sciences. It's the plant that is not a plant anymore, but a system to test all kind of fancy things on. You see here 3 variations, Colombia and so on. And we have given, well, not me, people have given them different levels of salt stress, 0, 100, 150 and 200 millimolar of salt. And you see that while these control plants seem rather happy, as soon as you start giving them more and more salts, especially this accession here in the middle is really starting to, well, there's not a lot of growth anymore. The growth is hugely retarded. They also they start getting yellow, they lose chlorophyll and so on. You see the same things here. The more salt you give, first of all, you have for the first set of concentrations, you see no effect on the time point where the little root appears, radical emergence. And then you see that a number that the roots either do not appear at all or will appear later and so on. You will see that plants start to have slower green cotyledons and so on. There are also plants that are tolerant for stress, for salt stress. And in this case, this is an older paper, already 2001, but at least you see here healthy Arabidopsis on no stress, no salt. Here you see not healthy Arabidopsis at 500 millimolars of salt after 10 days. You see these plants are dead. These are zombie plants, walking dead. Whereas here you have Telugiella halophila, which is a salt-loving plant, which does not grow very good when there is no salt available, and they grow very well on half a molar of salt. Even after 10 days, they look very green, very happy. Another effect is temperature. You see here that you have here 1, 2, 3 different varieties of hot pepper plants, Capsicum annuum. These are control levels after 34 days. And here after 75 days, again, even after 75 days, even the small one already bears fruit. Also, this one already bears fruit. Now, if you look at the plants that were treated with heat, that were grown at 10 temperatures more, then you see, first of all, they stay smaller. And secondly, well, here and here, these ones delay their fruiting response. And apparently this one has accelerated their fruiting response, which is also a possible response in the plant world. So temperature, stress changes the way this plant behaves. We also have biotic stresses. Biotic stresses are bacteria, viruses, beetles, herbivores. Even a cow can, for some discussions and some definitions, be called biotic stress. We have here a bacterial disease, we have a virus, we have here the beetle, and then we have different forms of fungi that all grow on the same maize plants. And here you have ergo, which is, as you may know from your natural history, also the source of lysogenic acid. And if you treat that a little bit more, you have LSD. It's a completely, nearly natural product. Again, there we see that if you confront plants with a biotic stress, in this case, it's a bacterium, Xanthomonas, with which this is, again, healthy Arabidopsis. And these are plants that were infected with this bacterium Xanthomonas. You see that these plants again start to die off, or their vegetative state starts to die off. And what you get is maybe accelerated flowering. And the plants just say, oh, I'm going to die. The only thing I can still do is make a lot of seeds, distribute the seeds, and then I can die off and my progeny will have to fend for themselves. I'm going to jump over this and maybe come back to that later. An important concept that has been found in stress is, well, all of these things, all of these forms of stressors, they all get to a kind of a similar physiological condition later on. And that's always one of the bigger questions in stress. That is that you have tens of possible causes of stress, but the outcome, the way the plant responds, both on a molecular level and on even a more macro physiological level, they are more or less alike. There are always a couple of specific responses. I mean, the plants deal with salt in one way and with metals in another way and with temperature in another way. But there is also always a kind of a general basic way that plants treat with stress. And that is the way using reactive oxygen species. So when we talk about stress, actually at a certain moment we will start to talk about oxidative stress. Plants are using oxygen, just like we are using oxygen, and because of the stress, either the plant is forced to create what we call reactive oxygen, or in some instances, again, as we do ourselves, plants are forming these reactive oxygen species voluntarily. They are creating reactive oxygen species in order to fend off the stress. Now, always a little sidestep. The fact that reactive oxygen species are formed, so it's a more dangerous form of oxygen, is a consequence of the fact that we all use oxygen. And let me first bring that to your mind. Why do we use oxygen? Well, there's a very simple reason that in biology, we also follow chemical laws. And this is a basic law in which a different chemical reaction produces energy delta G. And I'm sorry, I switched these. It's delta G should be here and delta E should be there. I switched these around. So delta G is minus NF delta E. And the point is actually that the bigger the difference in potential between your electron donor in your cell and your electron acceptor outside of your cell has to be as large as possible. If this delta, if that's as large as possible, then your energy difference will also be as large as possible. So depending on which electron acceptor you use, the more energy you will start to suck up from the environment. In this case, oxygen has a very high redox potential. So given the fact that in the cell itself, we always use NADH or NADPH, minus 330 millivolts redox potential. Oxygen has a redox potential of plus 770 millivolts. Because that's very large, we get more energy. There's a number of bacteria that start to use sulfates or nitrates or iron, but in all of these cases, the energy yields of these reactions are a lot lower. But why should we not use oxygen? Because oxygen normally goes to water. That requires a transfer of 4 electrons from oxygen to water, and also from water to oxygen. Now, in some instances, when these electron transfer reactions go wrong, then we start to form first superoxide. Superoxide is a radical which is much more reactive than plain oxygen and may be formed during these reaction chains. The second one, if we give oxygen 2 electrons, is hydrogen peroxide. And you all know what that does. It's disinfectant. It kills off a number of living cells. If you pour it on a wound, you can use it to disinfect the wound. That's because your H2O2 starts to attack your proteins, your membranes, your DNA. Let's take another electron. So we go for 1, 2, 3, and we end up with hydroxyl. Hydroxyl is something that, for example, in water purification systems, we use on purpose, again, to kill off a lot of unwanted bacteria and to degrade, very hard to degrade organic components. So that's when we use a Fenton reaction. I put a number of the reactions for Fenton here. This is a typical set of reactions that we use when we want to disinfect water or sanitize water. So that's the oxygen problem. So what happens now? We have our stressor. The plant reacts either because the normal reactions in the mitochondrion and the chloroplast are affected, so they start producing more reactive oxygen species. In other instances, the plant is actually starting to produce reactive oxygen species on its own. And well, the consequence is always, that's the basic hypothesis, well, we have then protein damage, we have DNA damage, we have membrane leakage, and that causes a general feeling of distress in plants. Plants get into trouble. Plants also defend against oxidative stress. One of the ways is by creating a lot of antioxidant components. I mean, you all know vitamin C, ascorbate. You've eaten it this morning, I guess. You had some orange juice, or you ate maybe an apple, or, well, that's ascorbic acid. Everyone eats tomatoes or bell peppers. You get lycopene. It's an antioxidant. Maybe you ate walnuts, then you had tocopherols. Maybe you ate grapes, then you had resveratrol, or even tocotrienol or tocopherol. You had broccoli or other cabbages for dinner. You had glutathione, you had beta-carotene, you had quercetin, you had capsaicin in your hot peppers. So every plant contains a number of antioxidant components. And those are there for the plant to combat, again, the oxidative side, the reactive oxygen species. And by the way, we also need that to combat our problems with reactive oxygen species. And I give you here, very briefly, a number of different things you would like to have during lunch: a glass of red wine, a cup of tea, an apple, an onion, orange juice, beer, or white wine. And then you see what you get in terms of equivalent of vitamin C. Lots of antioxidant components that you eat and drink the whole day long. Even chocolate, I'm Belgian, even chocolate is said to contain a number of good antioxidants, if you eat the very dark version, of course. Okay, and now we go to morphology, because that's what you're waiting for. First of all, stress can change the rate a plant divides. This is a root tip of a maize plant. And even already in 1995, I used this as the basis for my PhD application. Even in 95, we already knew that antioxidants can regulate the rate of cell division in plants. There was a whole model that always starts with the plant hormone auxin, because it does everything. Auxin would activate an enzyme that we call ascorbate oxidase. Ascorbate oxidase has long been an enzyme without a function. Here we found out that ascorbate oxidase oxidizes vitamin C ascorbate to its completely oxidized version, dehydroascorbate, and then blocks the cell cycle in the quiescent center. That's this zone in a root tip that has no or very, very, very slow cell division rates. Second, that's a paper actually that I was involved in already. Stress will also induce different ways of differentiation. For the non-biologist, differentiation is the process by which a cell acquires a different function. You are not born, well, you are born with a functional liver, but your embryo is just a non differentiated cell. So along your development, your body has a number of cells that at a certain moment decide, oh, we will be liver, we will be kidney, we will be brain. Same for plants. Plants have one extra thing. Plants can also go back. Even if you take a fully grown leaf, you take out a number of cells from that leaf, you can make these leaf cells grow back into even a root cell, even a shoot, whatever. And what we found is if we took leaf cells from an alfalfa leaf and we gave them a low level of hormones, then these plant cells would balloon up, become just larger cells. They would expand, they would grow. If we gave these plant cells a high level of auxin hormone, they would become a very compact cell that looked like a non-differentiated cell, a plant cell that we could give a new function later on. And then the trick became, if we gave the control level of auxin, and instead of high auxin levels, we gave them a form of stress, iron stress, salt stress, and so on, we could again push them to become non-differentiated cells. We could even play a bit with different forms of these antioxidants. If we gave them high amounts of glutathione, we would push them again to differentiated, non-differentiated cells. If we gave them high amounts of dihydroascorbate, the oxidized vitamin C, we would push them to become these cells. Again, what's the main message? Stress does not only change the rate a plant grows, it also changes what a plant cell becomes. And then we started to read a lot of papers. We found this ourselves. And we started to say, hey, apparently there are also macro physiological effects and macro morphological effects. So again, you see Arabidopsis, and you see here that we gave our Arabidopsis plants up to 100 micromolar of copper. Or we gave them even more stranger components that were bound to induce an antioxidant, an oxidative stress level. And we gave them actually components that started to produce reactive oxygen species on their own. And then we saw this, a little monster here, and we saw this little monster here. And we start to think, yeah, we see some kind of a pattern. We see a pattern that says, look, plants are stopping to have elongated, sorry, I'm going to go back. Plants stop to elongate either their petioles and their shoots, or they stop to elongate roots, and they start to grow more lateral roots. Here one, here one, here, here. Just to be clear, again, this is still the healthy Arabidopsis plant. These are the little monsters that we created. And then we continued our literature search. And the comment I always make here is, sometimes we are so obsessed with putting things in graphs and in numbers that we forget to look at, probably wrong place to say that, the general shape of things. We had to look, I think I dug through about 200 papers to find five that showed us what a plant was looking like under stress. For example, here, this is a maize plant where we gave them, they gave them a control medium, and this was a medium with high, actually not that high for a plant, rather high amounts of aluminum, right? And you see again, especially the root, stops elongating, makes a lot of very, very small side roots, but does not make very long, does not let them grow a lot. We looked at phosphate unavailability. If you give less phosphate to your plants, your roots become very short and you start again to form these little side roots. And then we even looked at, very bad picture, I know, but that is how we got it from the paper in 2004 paper. We even looked at elongation stopped and root hairs getting elongated by massive dose of gamma radiation, again, probably something that was caused by the presence of reactive oxygen species. From all these monsters that we found in literature, we came up in 2007 with a kind of a general model. That is that you have an interplay between stress, the hormone auxin, the stress hormone ethylene, reactive oxygen species, and something that we called the redox state of a plant. Well, actually not us, but that will affect division, elongation, and differentiation. And that will then cause a difference in general habitus of the plant. This is the control root. This is what we dubbed it SIMR, stress-induced morphogenetic response, SIMR. And this syndrome, it said, well, first characteristic of the syndrome is you have an inhibition of elongation of your root. You block cell division, your cell meristem stops dividing. And then you have a redifferentiation. So you, in the root, but away from the tip, you start up cell division activity again. And there you have formation of lateral roots. This was, for us, this was a very important point. If we only have inhibition of elongation and inhibition of cell division, then stress would mean you damage the plant. Here we say, no. We don't really damage the plant. We just redirect, or the stress redirects the activity, because here in your lateral, you have more lateral roots. So the cells are not just damaged beyond the possibility to ever divide again. No. They start dividing just on a different place. You change the general morphogenesis of the plant. And that's why we said, no, this is a very different response. Actually, paper has been cited more than 1,000 times. Ever checked how your papers were cited? You will find out that 1/3 of your citations are actually people that have not read it. Oh, yes. And then you hope that the other 2/3 at least have read it. Okay. We went further on that, because this actually means stress does not have to mean damage. If we say stress blocks plant growth, if we say this, then that's a very anthropocentric and maybe egocentric view of the plant biologist, because it means that we know what a plant wants, or we think we know what a plant wants, have lots of leaves, whereas the plant says, no, I don't want to have a lot of leaves. I just want to survive and continue to grow and not be disturbed anymore. So maybe it's a kind of the plant biologist that is brainwashed by the need to be as efficient as possible, meaning one plant, more growth, whereas the plant says, no. I'm very happy to do what I want. We even came up with a more complex idea about that. Again, you can read this. This is the one. The before here is the basic idea of how people think about stress. You have a stress, you have the reactive oxygen species, the antioxidants play with it, and then you have damage, and then you have the death of the plant. That's what I just explained. We said, well, maybe when a plant is subjected to any environment, then these reactive oxygen species and these antioxidants together are going to form a kind of a general balance inside the cell, which we then have to call the cellular redox status. And the cellular redox status will then trigger maybe DNA damage, or maybe really programmed cell death. Plant chooses to die, but maybe also different ways of development, maybe different gene expressions, maybe, and so on. So you have maybe just the plant wants to do something different with its life. So let's go to a more plant-based definition now. And I think you will all agree with me that Confucius made a very valid point when he said you have to use the proper terms and you have to define them properly. Otherwise, you do not understand what is meant or you do not observe what is meant actually. So we came up with this. Stress is something that is a way of a plant to adapt to life, to make sure that it stays alive. And stress, and I again quote Hans Selye, stress is the spice of life. Death is not caused by the presence of stress, it's caused by the absence of being able to feel stress, because then you can't adapt and then you will die after all. This is a very recent publication. I just put it in here because this starts to explain how stress may be able to redirect the direction of growth by creating different gradients in reactive oxygen species. Again, this is very recent, and I have no idea whether that can bring us towards an understanding of our SIMR concept. And then I will end with this model or our definition of stress. And I would say, look, a plant, even a plant that is growing in what we would call a stressful environment, if the plant, what the plant actually wants is that the inside metabolic state is in equilibrium with the outside environmental state. And sorry?
No, please.
Okay. So if your stress is light, then we call this, and we call this again with Lichtenthaler and Selye, we call this what we eustress. A light stress, that's the stress that actually pushes you just to become more in harmony with your environment. Your eustressor is only slightly pushing your outside balance away from your inside balance, and what the plant will do is acclimatize so that it will finally be in harmony again. Eustress is simply light adaptation. For the people that want to have a parallel with physics and engineering, call this an elastic response. The plant gets pulled away from its ideal position and will go back to that same state again. Maybe it has changed the way it grew a little bit, but it keeps on growing happily ever after. If the stress becomes sturdier, stronger, longer, we call it distress or distress. And then your plant will either die a little bit, maybe lose some leaves, maybe be a really, really strange monster. I think I've shown you a few. And you will never get the plant to completely live in harmony again. So you'll end up with plastic responses. And once you cross a certain threshold, there is no way of going back at all anymore. And then the plant will soon collapse and die. Now, I've already had a discussion with Johan about whether that could be linked to Rene Thom's catastrophe theory, but it will require a lot of thinking and chewing on the subject before I can really get that into words, because it's a very hard way. To conclude, stress conditions are important drivers that will help a plant to perceive the outside environment, to harmonize itself with that outside environment, and by that way optimize, not maximize, growth and development. And maybe the difference between optimization and maximization, that's maybe the best lesson we can learn from a plant. Thank you very much.